सेल डिवाइड होने से पहले इतनी तैयारी क्यों करता है? Seriously, think about it. I want you to close your eyes for a second. Imagine a cell not as some tiny microscopic blob, but as a giant, sprawling, futuristic mega factory. This factory is totally self-sustaining, it's humming with energy, and it's gearing up for its ultimate purpose, cloning itself perfectly. But before that spectacular grand finale of cloning actually happens, well, something wild is going on behind closed doors. It's honestly mind-blowing. You'd think the actual splitting apart would be the main event, right? But no, this biological factory spends almost its entire lifespan just getting ready for it. Here is a massive truth bomb for you. The interphase, which is that preparation phase between two successive divisions, literally takes up more than 95% of the duration of the cell cycle. To put that into perspective, imagine a human cell cycle takes about 24 hours. That cell is basically spending over 23 hours picking out its outfit, doing its hair, getting ready for a party, and the actual party, the M phase, lasts for only about a single hour. Now, this timeline varies wildly, depending on who we're talking about. Human cells, yeah, they take their sweet time, averaging a full 24-hour day to complete just one cycle. But yeast? Yeast is like the sports car of the microscopic world. A yeast cell speeds through this entire factory process in just 90 minutes. It just goes to show how much this duration can change from organism to organism. If we look at the factory blueprint, you can really see how massive the interphase is. It spans all the way across G1, S, and G2, absolutely dominating the cycle. And then right at the very end, there's just a tiny slice for the M phase, which is where the actual nuclear and cytoplasmic division happens. So the factory is locked in prep mode almost the entire time. Because of this, historically, textbooks called interphase the resting phase. Sabse bada jhoot. I'm telling you, it is the biggest lie in biology. The cell is absolutely not resting. Actually, scratch that. It's a completely chaotic, hyperactive period where the cell is undergoing intense growth and highly coordinated DNA replication. Calling it a resting phase is literally like calling a kitchen during the dinner rush a resting room. All right, let's dive into phase 1. G1 phase, the growth zone. Walking into this zone, you'd see bustling conveyor belts and just sheer metabolic overdrive. The cell is working overtime here, continuously growing in physical size and building up infrastructure. But, and this is a massive but, there is absolutely no DNA replication happening here. The central blueprint hasn't even been touched yet. The factory is basically just expanding its walls and hiring more workers. Next up is phase 2, the S phase, the glowing DNA copy machine. Once the cell is big enough, the heavy vault doors open. Notice how perfectly coordinated this process is in animal cells. While the DNA is being painstakingly replicated deep inside the protective vault of the nucleus, out on the main factory floor, meaning the cytoplasm, the centriole is simultaneously duplicating. It is a perfectly synced double-duty preparation. All right, guys, major NEET alert here. Pay close attention because this specific phase is the ultimate trap that examiners love to set on medical entrance exams. Seriously, students fall for it all the time. So here is how you avoid the trap. Let's memorize this together with a golden rule. Ready? Amount double hoga, count same rahega. The amount of DNA doubles, so if we start with 2C and G1, it blasts all the way up to 4C after the S phase. But the chromosome number, it stays exactly the same. If it was 2N before, it remains 2N after. Amount double hoga, count same rahega. Do not let them trick you on this. Moving right along to phase 3, G2 phase, the final preparation room. Surviving the S phase trap, we step into the final preparation room. Here, the focus shifts entirely to the armory. Proteins are heavily synthesized in preparation for mitosis. The cell is building the specific protein weapons and armor, like spindle fibers, that it's going to desperately need for the actual physical division. All the while, cell growth is still steadily continuing. Now for a quick detour to phase 4, the quiescent warehouse, or G0 phase. Wait, freeze the factory for a second. What happens if a cell decides it just doesn't want to clone itself? Because, not all cells are constantly dividing. Adult animal heart cells, for example, exit the G1 phase and enter this inactive quiescent stage called G0. But remember, they aren't dead. They remain metabolically active, doing their daily jobs, but they just no longer proliferate unless they get a specific distress signal, like an injury, calling them back into action. All right, time for a pop quiz. Let's do the onion root tip challenge. Our factory tour is almost over, so let's see if your brains are still in the metabolically active phase. Here is a classic mystery for you. You have studied mitosis in onion root tip cells. It has 16 chromosomes in each cell. Can you tell how many chromosomes the cell will have at G1 phase, after S phase, and after M phase? And what about the DNA content if it starts at 2C? Let's crack it using our NEET alert rule. Chromosomes?
Well, they don't care about the S phase at all. The count remains the same, so it's 16 in G1, 16 after S in G2, and 16 after the M phase. But the DNA amount, that starts at 2C in G1. Then the S phase DNA copy machine doubles it up to 4C. It stays 4C through G2. Finally, after the cell divides in the M phase, that 4C is split evenly back into 2 cells, dropping right back to 2C. Boom! Mystery solved.
So the factory is fully prepped, the DNA is copied, the proteins are forged, that long 95% wait is over, and the giant vault doors to the M phase are slowly grinding open. Ab shuru hoga asli battle, M phase or Prophase. You absolutely will not want to miss what happens when this factory actually tears itself in two. Will it survive the split? Keep learning with our next explainer, and I'll see you in the battle zone.